So on behalf of Dr. Joyce and John and the rest of our colleagues, allow me to again welcome you, warm welcome to round 4 of our CARPHA COVID-19 health rounds entitled Laboratory Diagnostics and Patient Immunity. My name is Sasha Wallace-Sankar Singh, the Biorisk Manager here at CARPHA, and today I'll be your moderator. So in terms of welcoming you, I just have a few brief remarks for housekeeping for the webinar and how it's going to be going today. It's approximately 2 hours and we will have one main presentation. So during this time, we ask that your microphones remain on mute. There will be no enabling of the video. And we're going to be asking you to use the chat box to enter your questions. While we have a question and answer session at the end, Gonzalez-Escobar, and by way of introduction, Dr. Escobar, who many of us have worked with over the last few years since he's joined us here at CARPHA, is actually coming to us from Colombia. He's actually born in Colombia, but spent his waking years in Venezuela, where he completed many of his degrees, including his medical degree, his diploma in public health, as well as his PhD in biological sciences. And many of us know that his major area of interest has always been in virology and particularly hemorrhagic viruses. Something that we didn't know about Dr. Gonzalez-Escobar is that in 2006, he developed a new PCR method to diagnose Venezuelan hemorrhagic fever. And we would know that this is a risk group 4 arenavirus, and it produces sudden hemorrhagic episodes in rural areas in Venezuela. So Dr. Gonzalez-Escobar comes with a wealth of experience as a doctor both in the public and private sectors. And in 2016, he joined us as the virologist, and now he serves as the head of laboratory services and networks here at our CARPHA Medical Microbiology Laboratory based in Trinidad. On a side note, Dr. Gonzalez-Escobar is one of those persons you can throw any language at and he'll be able to answer you. He's fluent in Spanish, English, Portuguese, Italian, and Neapolitan, with some basic knowledge he claims in French and German. So there's still more that we're learning about Dr. Gonzalez-Escobar, but for now, I will ask Dr. Escobar to share his screen and take us through laboratory diagnostics and patient immunity for COVID-19.
Good morning, everybody. Thank you very much, Sasha, for those words. I will share my screen right now. Okay. Okay. Can you all see my screen?
Yes, we can, and it's in full screen mode, so you're good to proceed.
Thank you very much again. Again, good morning, everyone. Thank you very much to all for joining us this morning. We will be discussing some very important aspects on the laboratory diagnostics of COVID-19. And we will also have some words for the discussion of some relevant aspects of the immunity and how it relates to some laboratory testing procedures. It's an honor and a pleasure for me to share these thoughts this morning with you, and I hope that you all will find this information helpful and, of course, interesting. So the objectives of these presentations are to provide current information on available laboratory diagnostic options and to provide information on what is known about patient immunity to the severe acute respiratory syndrome coronavirus 2 virus, or SARS-CoV-2. A brief outline of my presentation today. I will be talking about the status of the COVID-19 pandemic in numbers, some words about the origins of the SARS-CoV-2, the different laboratory diagnostic platforms that we have so far available in our region, what we know about patient immunity, some words also with regard to the evaluation and selection criteria of diagnostic kits, some considerations for the future, and of course we will have a session of questions and answers. This presentation is actually divided into 3 different parts. So at some point in time, and I will ask Sasha to once the opportunity arrives, to remind me that I have to pause and to let you take a deep breath, and probably you will be allowed to make some questions, and then we will restart with the following point. So Sasha will be in charge of pausing me, and she will be moderating this part of the presentation. Okay, this is the current panorama, the current picture of what we have with COVID-19 worldwide. These are numbers as of Monday, 21st September. Globally, we have more than 30 million confirmed cases, 206,000 from the Caribbean, in 33 countries and territories or areas, 120,000 recovered, and more than 3,500 deaths recorded. So of course, this situation is evolving every week. So by this week, we have different numbers, but this is just to give you a general picture of the situation. In CARPHA member states, as of September 16, CARPHA Medical Microbiology Laboratory received from 17 CARPHA member states, 22,788 samples, and 11.1%, 2,500 plus, were tested positive since the start of the outbreak. About the origins of SARS-CoV, this is a very nice picture of what has been the situation with different pandemics and different viruses over the last century. And you will note that in 1918, the Spanish flu caused approximately 50 million deaths. And that number was almost 3% of the world population. And again, 1.5 million deaths in the 50s of the last century produced by the Asian flu. Some others, like in 2010, the pandemic flu caused by H1N1, 300,000 deaths, but this time it was less than 0.004% of the world population. So the situation has improved in terms of the percentage of the population affected with regard to the deaths. But what is interesting here is that if you see the time that separates every pandemic from the previous one, we are talking about something like 40 to 50 years to face these problems. This time, from 2010 to 2020, it was really 10 years and not for the pattern of 40 to 50 years for the past pandemics. So what has been thought about the origin of this virus and the generalized route of transmission of the epidemic zoonotic coronavirus? Well, we have heard about the different theories about how this virus evolved and the change or the shift to different species, including the human being. So apparently the virus was circulating among bats first, and from time to time was transmitted to other species, mostly mammals. And the intense interaction between the human beings and this animal species produced at some point in time the capability for the virus to infect human beings and to produce disease. This has been the frame the frame of most of the respiratory transmission cycles produced by viruses that originally infected only animals and then crossed the barrier and started infecting human beings. So of course, being a respiratory virus, the possibility of transmission and the capability of being transmittable is very high. And of course, it occurs at a community level. And this time, this has been favored by the fact that we are in close contact, mass gatherings, and we have a high rate of mobility across the world. So as never happened before, the virus can be now in some point of in the planet, and then 24 hours later can be in the antipode of that point. And of course, this is being facilitated with the air transport and sea transport and the intense mobility in the modern times. Other aspects of this transmission is that now the hospitalized individuals with a diagnosis of COVID-19 disease can produce nosocomial transmission, can affect healthcare workers, and can also infect other patients in the wards. There have been produced a very important number of documents by almost all the international health organizations like WHO, PAHO, CDC, all the public health agencies, including CARPHA. So you will see that this presentation, some of the aspects of this presentation are based on several documents from the World Health Organization, the Pan American Health Organization, and also on our updates, our CARPHA laboratory updates produced from the beginning of the outbreak to date. Let's check one moment about the humoral immune response to SARS-CoV-2. Sorry. This chart shows you the dynamics of the production of several antibodies, especially IgM and IgG. I'm sorry, but IgA is not included in this chart. But you will see that we have here three very well-defined periods. One is the window period, one is the decline, and the convalescence. So you will see that, for instance, the first immunological marker produced is the IgM that is produced after the interaction of the virus with the cells. So we can first detect basically antigens, viral antigens, and viral RNA from the window period and on. And then you see that IgM becomes available approximately 7 days after the infection occurs. And the IgM is considered to be present in the bloodstream up to the day 21 after the onset of after the infection, and it reaches some undetectable levels at that time. Then the IgG, and the IgG is present from the day 14 and on, but the actual levels of the IgG and how these levels last, how much these levels last in the bloodstream of a patient infected with SARS-CoV-2 are a question now that is on research, because it is not known how protective these IgG titers are, and if these IgG titers provide long-term immunity. This is with regards to the humoral immune response. But also we have a very, very complete spectrum of other immune responses. And most of the immune responses to SARS-CoV-2 have been obtained from the previous research with MERS and SARS-CoV. I know that this is a very complicated chart, but I will tell you that basically the virus interact with the cells at the respiratory system, and the virus has basically two different ways of entering the cells. The first mechanism is a direct fusion of the membrane, the cytoplasmatic membrane and the viral membrane, which releases directly the RNA into the cytoplasm. And the second mechanism is a receptor-mediated endocytosis. In both mechanisms, the cell detects the presence of the single-stranded RNA and the double-stranded RNA. And this process activates some sensors. In this case, the two sensors are the REACH and the MyD82. These two sensors produce a downstream for the production of several factors like the IRF and the nuclear factors. These nuclear factors produce the transcription of several and different pro-inflammatory proteins like cytokines, interferons, ISGs, etc., etc. Also, some viral proteins can induce the production of a process called pyroptosis, which is a way of programmed dead cells mediated by the use of the interleukin-1 beta. This is basically the viral recognition by the innate immune system. But there is also another process going in parallel that produces the factors that or the characteristic immunopathology of SARS-CoV-2. This, the immunopathology of SARS-CoV-2 resembles what was seen again for SARS and MERS. Some recent studies have determined that there is an increased production of interleukin-6, 10, and the tumoral necrosis factor. And also there is a problem with the production of T cells, and T cells are not competent. And this is this part of the chart. What produces this incompetency of the T cells is a high level of production of cytokines. And then this cytokine storm produced the hyperinflammation seen, especially in the lungs. So this is a summary of what is happening at the molecular level in the respiratory tract cells. Sasha, you may indicate to me if I have to pause here, or can I go?
You can go forward, Dr. Gonzalez. I do not see any questions at this time in the chat or in the question and answer box.
All right, noted. Thank you. So let's talk about the different laboratory diagnostic options that we have right now. So we'll start with the serology, and I intentionally left the PCR for the end of the presentation. So there are several reasons why antibody detection tests, which detect IgM, IgG, and IgA, cannot even be considered at this point in time as diagnostic tests. Number one is that there is a strong possibility of cross-reactivity between in this test between SARS-CoV-2 and other coronaviruses. As has been seen for other viral families and genres, the cross-reactivity represents a problem of false positive results. Number two is that the detectable levels of antibodies only appear after 7 days from the onset of symptoms. So a negative result in the first 7 days is not useful to rule out the presence of the case. Thirdly, the presence of the antibodies is not synonymous of protection against SARS-CoV-2, because we now know that there are several different subtypes of antibodies, and not all these antibodies are neutralizing antibodies, so they are not really protective. And finally, and I consider this the most important aspect, is that the actual sensitivity and specificity data of these tests using samples are very limited and need further evaluation to determine the usefulness in the context of the SARS-CoV-2 pandemic. So we have, in one side, a very good test, which is the ELISA, but with the possibility of cross-reactivity, with no detection of antibodies during the first week after the infection or after the onset of symptoms. We have the possibility of reaction with non-protective antibodies, and then lower sensitivity and specificity when you compare to the PCR. Then we have the second line of test, which is the rapid viral antigen detection test, or RADT. The purpose of this test is to detect the viral proteins that are excreted during the first days after the onset of symptoms. To give you an idea of what can be similar to this, for dengue, for instance, we have the normal or regular detection of some antibodies, but we can also detect one protein with one different test, which is the NS1. But this is more or less the same principle. So during the infection, some viral proteins are available, and there are tests for the detection of these proteins. Although these tests have been effective in some public health settings related to SARS-CoV-2, they also show lower sensitivity compared to PCR. PAHO has published a very useful document on laboratory testing, and in that document, and I quote, "A negative result at any stage of the infection should not be used as a criterion to rule out a case, etc., etc. So what is this telling us? Despite of the usefulness of this test, it can be used to rule out cases. In a very recent report also, it was shown that the sensitivity of this test can drop down to 60 to 77%, depending on basically the level of the day of when the sample was taken. And when you compare to the CT values in the PCR, the lower the CT, the higher the sensitivity of the test, because there are more viral proteins available. So this is a very serious matter of concern, that if the antigen tests are used as a first line of diagnostic test, it can have, as a result, it can show very, very important number of false negative results. And also, PAHO has recommended that the RADTs are only used for lower priority testing. So in that document, you can go to that document. It was published in July. You will see what, and at the right side of the slide, you will see the algorithm for the use of this antigen detection test. And well, you will see that when the antigen is positive, it can confirm COVID-19, but whenever the antigen test is negative, it has to be confirmed by RT-PCR. So in general, as a general rule, these tests are only recommended for symptomatic patients during the first 5 to 7 days after the onset of symptoms. And of course, all all negative results have to be confirmed by RT-PCR. In the study that I was referring to previously, with the level of sensitivity of several antigen detection tests, you see here this part of the results in that report. You will see, well, that the level of sensitivity for this test was actually related to the CT values. So when you had a CT value less than 18.57, the sensitivity was 60 and 77.5% for one test. When the CT values were higher than 18, the sensitivity dropped down to 45 and 60% for both approaches. But when we do the evaluation of what happens in real life, and these are data from the CARPHA Medical Microbiology Lab from August to September, we did the evaluation of 1,618 samples received. Only 19% of the total positive samples had a CT value less than 20, but the rest of the 81%, the CT values were actually higher than 20 or higher than 30. So in real life, what we are seeing is that the samples that are coming to our labs have, in general, a very high CT value, and therefore very low viral load, and this can complicate the use of the antigen test because their sensitivity depends on the level of the CT in the samples. So let's talk about the PCR. It's time to talk about the PCR. So far, PCR is the only method considered the gold standard for the detection of SARS-CoV-2. Sensitivity and specificity greater than 94%. We all know about this. And we also know that PCR is not exempted of problems or disadvantages, and that the sensitivity of the test can be affected by the quality of the sample, the incorrect transportation of the sample, and leaving the sample at high temperatures for long periods of time, the flaws in the RNA extraction procedure. If the procedure is not followed correctly, it can produce less RNA and level, and of course a false negative result. Poor nucleic acid concentrations, the presence of inhibitors of the PCR enzymes in the sample, or inhibitors in the kind of swab or medium used for the transportation of the samples. Also, with the accumulation of mutations in the genome, in the RNA, it always exists the possibility that these mutations can reduce the sensitivity of the test, because the target regions will not be recognized by the primers. And of course, the process of an incorrect process of sampling. I will not go very deep into this, but this is just to remind you that we have several options as targets used for the PCR confirmation. Most of the accumulated experience for SARS-CoV-2 comes directly from the past situations with SARS-CoV and MERS-CoV. So now we have a plethora of different targets into the SARS-CoV-2 genome, like the RDRP or RNase-dependent RNase polymerase, the E gene, the N gene, the ORF, in several of its in different modalities. So we have. It's very common that we are now talking about what are you using for the detection. Well, we are using the E gene or the N gene, or different combinations. So especially some pre-prepared kits come with different combination of E gene plus RDRP, or E gene plus N gene, or E or N1 and N2 on N, or NR and ORF, etc., etc. I want to use this. This was taken from a private company, a private lab company in Europe, but I want to use this to highlight the importance of the PCR in the context of the SARS-CoV-2 situation. So see, for instance, that this company uses 5 different examples of 5 different situations, actual situations and field situations. So for instance, you will see here, situation number 1, diagnosis of acute COVID-19 illness in symptomatic patients. So if you go down through the algorithm, you will see the importance of the RT-PCR for the diagnosis of the acute COVID-19 illness. So situation number 2, the investigation of symptomatic, asymptomatic contacts and carriers. So again, RT-PCR has a role to play in this situation. For the follow-up of hospitalized patients, is now the rule that there's no need for specific tests, and there is no recommendation for that. But of course, for hospital discharge, and very important for healthcare professionals, this company in particular recommends the periodical RT-PCR test every 7 to 10 days. But this is just to highlight that in every single situation, practical situation, in the fight in the fight against COVID-19, PCR plays a very important role. Another question that we have related to the PCR is, do we need a cutoff for the CT value? And I'm bringing this because this has been asked to me many times from different laboratories. Many persons, many colleagues across the region and outside the region have participated in this discussion. So it's now a never-ending matter, but we need at some point in time to clearly define this situation. And I took this graph from one paper published very recently. The authors of this paper determined that with the evolution of the disease in days, the trend is to have less symptoms, and this lack of symptoms, or this decrease in the symptoms and the severity of the disease, is directly related to higher CT values. So this is giving you the first insight about apparently there is a correlation between the presence or absence of symptoms and the severity of the disease and the level of the CT value, which reflects the viral load. Secondly, a different group in Public Health England published this work a month ago, yes, and they demonstrated that with highest value of CT, the recovery of infectivity in viral cells is less and less whenever you go to a higher levels of CT value. In other words, the higher the CT value, the less infectiousness can be demonstrated in a cell culture. In this regard, I have to say that we have done the same, we have had the same experience in our laboratory in CARPHA. And so far, our data indicate that above a level of a CT value of 30, the recovery of infectious in viral cells is almost zero. And with CT values less than 30, you will get more infection recovery when you approach to values less than 20. So it's
Dr. Gonzalez-Escobar, can you hear me?
Sorry?
We lost you for a little bit. We could see you speaking, but for some reason, the volume there went a little bit. I don't know if you can repeat your point after the experience you just spoke about with CARPHA with respect to the recovery of cells.
Can you hear me now?
Yes, we're hearing you again. Thank you.
Okay, yes, I received a message that my internet connection was unstable. Sorry about that. Yes, in our experience, we have recovered infectivity in viral cells with CT values less than 30. More than 30, the recovery of infectiousness, of infectivity in cells is zero. And this is our experience. The experience in this work is a little bit different. So they are able to recover 30% in levels like 32, 33, and less than 10%, 8% with the CT values around 37. But in both cases, we now know that at least the recovery of infectiousness, the property of the virus being infectious in cells is dramatically reduced when you have CT values higher than some point in time, and at some point in time, and this cutoff value, despite of this is an in vitro situation, not an in vivo situation, it can be assumed that there is some translation to the in vivo situation. So this remarks the necessity of having a cutoff value for the PCR for SARS-CoV-2. And also we have to remember that for the other systems that we have available right now with the PCR, like the system for influenza A, B, non-influenza viruses, etc., there is always a CT value cutoff. In the same way, we have noticed that the current system of detection, regardless the level of CT and the shape of the curve, because remember that regardless the level of CT, the shape of the curve is one of the parameters to be considered to assign positivity or negativity. But we have seen a high level of subjectivity with this system. So there are situations like this one, the first two squares, and I'm showing you the plots, the PCR plots that we get in our laboratory. You can assign positivity here more based on the CT value, which is 36, but not for the using the amplification plot. And you see that in this case, in the second case, the same case is the positivity is assigned based on the CT value, but the curve is slightly different. Sometimes we, at the level of our laboratory, we call this like blips, not really sigmoidal curves. But the question here is are these blips reflecting a biological situation where viable virus is present in those samples, and is there any possibility of transmission? So by having a CT value cutoff, these questions can be addressed. And again, of course, we have some positive cases like you see in the two boxes below, where the CT value is pretty high, more than 37, but you see a more sigmoidal shaped curve. The same situation can be applied for negative plots. Again, these are CMML data, where sometimes you know how to react to this. So you have CT values around 37 with a very poor sigmoidal shaped curves, but you are getting a signal. So this has been a very recurrent question from all our colleagues from the public and private sector in the region. And at that point in time, my only response has been, we have to wait until further research demonstrates the usefulness of a cutoff value for the SARS-CoV-2 PCR test. So finally, for this, it appears that what we are doing is that we are reporting most of the cases within a total scenario, and we are including in these positive cases both the infectious SARS-CoV-2 patients and the non-infectious SARS-CoV patients. In the first case, we have patients up to day 8 to 10 from the onset of symptoms, which are really shedding infectious viruses. But in the second case, we are assigning a positive result to patients that don't really represent a risk in terms of transmission and a risk for different in the public health setting. So I think that we need to understand who is really infectious and who is not, and how this can collaborate to the situation at a very public health level in our countries. I will talk now about some other alternatives available in the market for the PCR, and we have this alternative as it's called the RT lamp. It's a system that doesn't use complicated devices. It's a color-based system that permits you to detect positivity or negativity based on the color of the reaction. RT lamp stands for reverse transcription loop-mediated isothermal amplification. This system has several advantages, but also, unfortunately, some disadvantages. It's a quick technique. It can produce results in 2 or 3 hours. Those results can be read using the eye, so you just will see the migration of the color, and you will be able to decide what is positive, what is negative. It's a simple and cheap method, can be done within the hospital laboratories, but can be done on different settings. LAMP can detect current infections of disease, allowing medical staff to determine who is currently infected and who is not. So it is detecting actually the presence of the antigens of the virus in those samples. Unfortunately, as I said, there are some disadvantages. This technology is newer than the RT-PCR. There is no such a large background, so it needs more and more research and of course more determination on the quality parameters and the usefulness in the clinical settings. The science behind this is a little bit more complicated than the PCR, than the normal or traditional PCR. For those patients who have already cleared the virus and are in the recover phase, the test is not really useful because you can detect the presence of the virus. So there is another disadvantage, which is the different levels of virus, the distribution of virus, which is different in different levels of the respiratory tract. So you can obtain false negative samples if you take the samples in sputum or nasopharyngeal swabs, and not from the lower levels of the respiratory tract. Sometimes there are problems with the cross-reactivity with other coronaviruses genomes. So the primers and the probes are not that specific than those used for COVID-19 or SARS-CoV-2. So it can be a problem sometimes. And also, the test cannot tell you if the person is infected with this particular COVID-19 virus. Finally, we have other alternatives, more or less available in some reference laboratories. One is the neutralization assay. It's a very good assay. It's time consuming, staff consuming. It's not an easy one, but this test can tell researchers if the patient has antibodies, and more importantly, if the patient has developed neutralizing antibodies. The test can be performed using blood, serum, plasma samples, and is majorly done using very sick cells. And there are other immunoassays like the chemiluminescent assays. They are not widely available, and but they represent an alternative for those laboratories who want to have a different or an alternative test for the detection of antibodies like IgM, IgA, and IgG. Sasha,
Yes.
please.
Okay. Dr. Gonzalez-Escobar, thank you. This is a good time, I think, that we can all take a little pause. There are a few questions and one raised hand. So I will go to the question and answer first, that box. And the first question we had was really, is there any link between being asymptomatic and having partial immunity?
That's again a very good question. Most researchers think that yes, there are some so far unknown immunological markers or HLA markers that can produce that can be related to the occurrence or not of symptoms and the occurrence of or not of a more severe disease. Yes.
Okay, thank you. So Dion Young, Mr. Dion Young, hope that has answered your question. And for others who might be thinking that, there's a second question here from Sam Williams from Turks and Caicos Islands. And he's saying, is that to say there are instances when this relates to when you're talking about the cutoff values? Are there instances where you would have a value above the threshold for the PCR reaction where you would still deem it negative?
That question came from Samuel Williams.
Samgeo?
Samgeo Williams. Oh, yes. Unfortunately, that is one of the things that has to be resolved, Mr. Williams. That we will have results above the certain level, and then you will see a nice and very well shaped sigmoidal shaped curve. But again, more research is needed to really link the presence of a signal above certain level of CT and the infectivity status of those patients. But as I said, it appears that the higher the CT value, the less the risk of infectious is.
Thank you, Dr. Escobar. And from Eric Carbon from Dominica has asked, good morning, Dr. Escobar. With respect to clients with low viral load, what methods were used to detect non-infectious versus infectious? So that's the first part. Second part says, also, are there any recorded confirmed cases where the virus is reactivated in a client to produce symptomatic disease?
Thank you, Eric. It's very good to have you here. First question, there is no method to determine the infectivity of a patient with a high viral load. I mean, there are some tissue culture and cell culture methods to get into that situation. So you can recover the virus from cells, and that is telling you that yes, this virus is infectious to this particular cell line. But from person to person transmission, there is no way to determine right now, at least not at the laboratory level. So you will need to, more research is needed to determine what is the real dynamic between in the person-to-person transmission. With regard to this, to your second question, as far as I know, I can't remember now any paper with the recovery of virus, especially at low CT values, and that can be assigned as a reinfection or as a different infection process. What is recorded right now is that patients can be shedding viral particles, subviral particles, and neutralized particles and also RNA fragments for several weeks and sometimes for months. In fact, I remember a case of a patient tested at CARPHA. That patient tested positive in early April, and then the patient recovered after 2 months of several positive tests. And I don't know if you remember that at that time, the norm said that any patient to be discharged needed 2 consecutive negative results in 24 hours. Well, this patient, after 6, 7, 8 weeks, this patient got those 2 consecutive negative results. Then the patient, 2 months after that discharge, the patient underwent to a surgical procedure. So as part of the surgical procedure protocol, he was retested for COVID-19, and that test came again positive. So the patient, if you get, if you make the count of the number of days, weeks, or months, we are talking about, in this case, of 4 months. But more than likely, the patient was just shedding non-infectious particles or RNA fragments from the lowest part of his respiratory tract. But I don't remember right now to see a reinfection with high viral loads. What I assume, and based on the previous experiences with other viruses, is that once you face a second reinfection, you will not develop the same disease at the same level in terms of the seriousness of the symptoms. And also remember that you have at that time immunological background against the virus. So the new virus or the new challenge will be, in theory, rapidly controlled by your immune system, mediated by the presence of neutralizing IgG antibodies, and also by the humoral factors and the cellular response.
Thank you, Dr. Escobar. At this time, I'm going to invite Marjorie Rivera. I'm going to allow you to speak so that you can ask your question, and then there's one other question that we have to answer in the question answers box. I'm going to encourage you, colleagues, to continue sending your questions here so we can answer it. We're coming up on the halfway mark. So Marjorie. Do you still have a question for us that you'd like answered? Please feel free to unmute. Okay. I think that we'll move past that with Marjorie at this point. And Marjorie, please note that you can always type your question in the chat box or the question answer box. So we go to the final question that we have here. And the question from Sani Van Kampen Could you tell us anything about PCR tests that use saliva, please?
Yes. In general terms, it is considered that the saliva is a very good sample, but it has shown less sensitivity when you compare with the nasopharyngeal swab. In fact, in terms of the sensitivity of the assays, in the first place, it is the nasopharyngeal swab, then the oropharyngeal swab, and then the other sources of samples like saliva, like the spit samples. And sometimes for some PCR methods, you have to dilute the saliva using different approaches, like the chemical or the temperature, the physical one, which uses temperature. Sorry about that. To make the saliva more accessible for the extraction. So these different alternative methods have demonstrated that they can affect the sensitivity, the overall sensitivity of the test.
Okay, thank you. At this point, we have a hand raised from Shireen Reed-Gordon. Shireen, I'm going to allow you to speak to answer your question. Please let us know where you're joining us from. Oops, I think. All right, there we go, Shireen. Shireen, can you hear us? Okay, at this point, I'm going to just ask Shireen if you still want to be able to ask your question, to put it in the chat box for us. Oh, you're able to speak. Excellent. Please go ahead, pose your question. Yes, Shireen, we're hearing you. Please go ahead.
Sorry about that. The hand was raised in error, I was saying. But I'm joining you from Kingston in Jamaica.
Thank you and welcome. Okay. Okay, Dr. Escobar, I think that's it for the questions at this point. So I think we can move forward to that last segment that you have for us. Remember, colleagues, please put your questions in the question answer box or in the chat. Over.
Okay. In this second part of the presentation, I want to show you some of the work that CARPHA and the CARPHA Medical Microbiology Lab have been doing during the last months as part of the response, of the emergency response to the COVID-19 situation. In June, we produced this document, the document that you are seeing on your screen. Is the CARPHA guidance for the evaluation and selection of diagnostic tests for the COVID-19 response. We firstly published this in May, but then we sent a revised version of the document. It's available in on our website, so you can go and get the document. And it's a very useful document that comprises 14 different parameters for the evaluation of the diagnostic kit. So whenever you are approached by suppliers and companies representatives, you can use this document to have a better idea of the capabilities and the potential use of those tests, of those kits. So these 14 parameters are the type of test, the sensitivity level, the specificity level, the evidence of other published or stated quality indicators like accuracy, precision, etc., the evidence of published peer review performance, the evidence of cross-reactivity with other coronaviruses, if the product has obtained any clearance from the Food and Drug Administration in the United States or any regulatory agency in the European Union, the evidence of the use for or endorsement from any international health organization or public health agency, WHO, PAHO, etc., the relative number of cost per test, the accessibility for procurement. You know that procurement sometimes, especially in our region, can be a challenge. So if that product is easy to be procured or not. If you need additional pieces of equipment to get your results, and how complicated are those equipment. So it's not the same to say, okay, we need a large piece of equipment in our laboratory with training, and we have to bring the technicians for the installation. Or on the contrary, this is just a all in a box test, and we don't know any additional equipment or any additional reagents. If you need a process of training to your staff, and if the test comes from a very well-known company, or on the contrary, it's a company that we have no clues about the reputation of the manufacturer or the company. So you will be assigning 0, 1, or 2 points. The matrix is very well explained. So you will have the opportunity to use this instrument as a very useful tool. And then, well, you will have different situations, and your results will be categorized into 5 different possibilities. So for category A, the kit shows a very good level of sensitivity. It can be used as a first-line test, or going down into the list, you will see category B, C, D, and E with different point levels, and then in different possible uses for those kits. And also there is one situation for antigen detection kits, but also for the antibody detection test or antibody detection kits. And again, the same applies for these antibody kits, antibody detection kits. Within the document, you will also find examples of the use of the instrument, 3 very concise and precise examples. So we presented in that document 3 alternatives as examples of the use of the instrument. The second thing that I want to refer to during this last part of the presentation is the effort that this laboratory has been doing with regards to the external quality assessment program for SARS-CoV-2. So far, we have sent more than, to be exact, 23 different panels to different laboratories across the region for both public and private sectors, and as a mean of verification of results in our laboratories in our member states. The response has been very, very satisfactory. We are very, very glad to see how our laboratories have responded to this initiative. And we want to invite again those laboratories. And again, I want to stress that this has been designed for both the public and the private sector, and to contact us and for those laboratories that are interested in participating in this program. It's a very good way to ensure that we are providing results with the highest quality possible. Finally, I want to mention some key considerations. The use of different tests, the use of different approaches, the use of technology will depend on the phase of the pandemic and the different phases within the first wave or second wave or third wave. Considering that within the waves, we have a period of for the introduction of the virus, then we have transmission in clusters, then we have community transmission, and we have the peak and the returning to the previous situation. So for each situation, we need to be smart on the use of the technology and the use of the test, and it has to be also related to the sustainability of this test during this pandemic. We have also to consider the use of different algorithms for different methods. As I explained, well, you saw an example of the use of antigen tests in some particular settings, not as a first-line test, and also the incorporation of the PCR into all these algorithms. Because as I said before, PCR is still the only gold standard for SARS-CoV-2. We extend our willingness to participate in the verification process through our external quality assurance program to all laboratories that are interested in this. And of course, we can assist countries and our national public health laboratories with the reception of 100 of your positive samples and 10% of your negative ones on a quarterly basis to follow up the quality of the results that you are getting in your laboratories. We have some challenges, of course. One of them is the immediate and continued access to suppliers and service providers, especially under these restrictions for mobility. We have challenges to bring people across and to bring technicians and to continue with the quality requirements for our devices and our tests. This situation, of course, will improve once those restrictions are lifted. The sampling strategy is always a point to be considered. It's not the same to take a nasopharyngeal swab or to take a serum sample or a urine sample or any other sample. So the sampling strategy has to be on hand and has to be related to the test and the approach that we are doing at one point in time. Of course, our normal recommendation continues to be the use of the PCR and continues to be as a very accurate sampling strategy, the use of the nasopharyngeal swabs. We have to have staffing considerations, which is always a challenge in the region, and the unavailability of personal protective equipment. In summary, I will state that PCR, of course, as I said before, remains the gold standard for SARS-CoV detection. There are other PCR variants and developments now on the pipeline, but more research is needed to determine the level of sensitivity and the comparison to the traditional real-time PCR. And the serology and the detection of antigens can be used in specific settings, but not as first-line test. We also need a sharper definition about the cutoff value for the SARS-CoV-2, and the effectiveness and the PCR results are more and more in close relation. We need more research on how this relation can be related to the person-to-person transmission and the role of the subviral particles or the RNA fragments or the viral fragments or neutralized viruses in the person-to-person transmission. The use of an evaluation instrument like the CARPHA guidance for the evaluation and selection of diagnostic tests is strongly recommended for laboratories and national health authorities to provide insights on the actual performance and usefulness of test kits. And as a very important tool, CARPHA Medical Microbiology Laboratory verification panels are available and for the evaluation of performance of public and private lab across the region. I want to acknowledge my fellow colleagues in the laboratory, Sumit Nathaniel, Risha Singh, to all CMML staff. Special mention to Sasha, who has been very supportive with this exercise. And again, I want to thank all the staff at the laboratory. They are not my staff, they are CMML staff. And I have to say that this is the more capable and more intelligent staff I ever had in my professional career. And with this, I thank you very much for your attention.
Thank you very much, Dr. Escobar, for that. We do have 2 questions, and I think your summary may have answered it, but the first one is from Dr. Giddings from Guyana, and he was asking, what is the recommended upper CT value for positives? So if you could answer that.
Thank you, Dr. Giddings. It's a pleasure to have you here. That's precisely the point of the discussion. We don't have a CT value to assign positivity. That's precisely the whole thing about the CT value. I could give you my personal opinion, but I don't want to confuse anyone, and I don't want to state a value right now because, as I said, we need an official recommendation. We have prepared a document with some recommendations with regard to the CT value. But of course, and I have to recognize that I've been a little bit hesitant on the publication of this document because we need more data to have a stronger recommendation about a CT value. But yes, that's precisely the whole thing about the CT value, that we need to determine a value.
Okay, thank you. And we have another question here about recommending the use of mass testing. So would you recommend the use of mass testing for COVID-19 to obtain a national prevalence estimate? And would there be a place for mass testing in outbreak management?
Okay, a mass testing?
Yeah.
I understand.
Widespread testing, mass testing of persons.
You mean a widely used in persons?
Yes.
Yes, of course. There are some precise indications for the use of some tests to determine, for instance, the prevalence, and that is precisely what is recommended for IgG, for the ELISA IgG. Those tests can be very, very useful for the determination of the prevalence into the population, within the population. So yes, you will see that if you want to use one particular test for mass testing, PCR is not the obvious choice due to the cost. So other tests which are really cheaper and can be where you can get results in less time and also with accurate results are obviously the choice, but it's not precisely the PCR. I would say that in this regard, the antibody detection test, like in serology, like the ELISA, is the more obvious choice.
Okay, thank you. From our friend Samjeo Williams from TCI, he's asking, once the CT value is published, would that CT value vary between assays and platforms?
That's a very good question. Definitely. We need to know the CT value, but we have also to consider what is our target. I mean, the CT value can't be the same if you're working with the E gene or the N gene or the ORF or any other. So it has to be specific for the target. I would say that is the first parameter to be considered, and then there would be some other considerations with regards of the type of the approach that they're using. But in the first decision that has to be made is what target are you using? So far, we know that the E gene is the most used target right now, followed by probably by the N gene, and more DRP has been decreasing because of the less sensitivity of this target when you compare to the E, the ORF, and the N regions. But yes, the cutoff value has to be linked to the target region that you are using.
Thank you, Dr. Escobar. There is another question from Kamli Ramsawak. What role does the LOD, that's level of detection, play in terms of the sensitivity of the SARS-CoV-2 virus for positive or negative samples?
Can you repeat again? I missed part of the question, Sasha, please.
What role does the LOD play in terms of the sensitivity for the virus in positive or negative samples?
I presume that Kamli is referring to PCR?
Yes, I presume that too.
Yes, yes, of course. The level of detection, when you go to, there is a foundation hired by WHO who is conducting these assessments for different PCRs and antigen and antibody detection tests. This you can go to the website. The foundation is called FIND. It's the Foundation for Innovative Developments or something like that. When you go to the level of detection of several different tests, you will see that some tests consider positivity when you reach CT values as high as 42, 41, 40. And of course, then you will see different level of detection for, especially for those closed systems like the Abbott M2000 and the FilmArray, not the FilmArray, oh gosh. Well, You will see the Abbott M2000 is the best of the examples that comes to my mind right now. So you will see different levels of detection. So you can't compare, for instance, what you see with a closed system and what you are doing with an open system, because they are two different approaches, two different, in many cases, the probes are totally different. So yes, each test has to be a LOD and has to be a cutoff.
Thank you very much. Colleagues, in the chat box, I have actually placed the web link for the organization for FIND. So you can click there and you can also get some further information. There's just a question as to whether or not this recording would be made available. We will review the recording at the end of this session, and we will be guided as to whether this can be shared externally in full. But do know that the presentation will be made available to all participants of this session as you have registered for the session. So we will confirm that the recording will also be made available as well. So thank you for your appreciation of the information. Okay. At this point, I'll ask again if there are any questions that persons have. At this point, I see persons saying thanks and good presentation. We've really appreciated you, Dr. Gonzalez-Escobar, in terms of being able to answer those questions for us. And so if there's anyone that would like to raise their hand or pose another question for us, we have a bit of time, just under 30 minutes, but we can also get towards the end of the programming for today. Okay. So at this time, I want to thank everyone for your rapt attention, as well as the questions that have been asked of us. I think they were very high quality questions as well. And for Dr. Gonzalez-Escobar for being able to answer those questions. As you can tell, we're still in the middle 6 months on learning more and more about this disease, about the way the virus works, and of course for our options that we have as laboratorians in terms of improving the platforms and the access for what we have available to us. At this time, I would ask everyone to note that you will be receiving a link in your emails tomorrow. However, I want to be able to share this link with you in the chat box for persons who may have joined late. I please ask you to click that link and provide your feedback to us on what went well and how we can improve. Of course, we want to remind you as well at this point that you can rely on CARPHA for all your information needs. We also have very high quality technical guidance documents available, and I've placed that in the chat as well for our COVID-19 page. We do ask you that you visit the site, that you go through the technical documents, and particularly the document on the evaluation and selection of diagnostic kits, which we think is invaluable for all of us to be familiar with and to use it as well. We know that this is something that is being asked of us more and more from suppliers and from others who want to introduce new products onto the market, but we need to ensure that we're following our process to ensure quality and safety of what we're doing. So from me to you, thank you for joining us for round 4, webinar number 4 of CARPHA Health Rounds. Look out for our upcoming health webinars and keep posted to our website. Dr. Gonzalez-Escobar, last word over to you before we sign off.
Thank you very much, Sasha. I want to thank everyone who joined us today. I hope that this was useful. We are here to assist. We are here to help you. You know that you can write us through the customer service at carpha.org, customer service at carpha.org. If any other question or you want to inquire anything else, feel free to do that. And also in our permanent role of as advisors for the region, feel free also to contact us whenever you have any situation or particular situation that you want to solve or any concern. We are here to assist. And well, your comments on this presentation and your suggestions will be more than welcome. And I look forward to having another session soon with all of you. And again, I thank you very much for your attention and for your kind comments in the chat. Thank you.
Thank you, everyone. Enjoy the rest of your day. Bye-bye from CARPHA.